Um, I heard that the test was easy, too easy, so the next one would be tougher. I'm not allowed to talk about it yet because not everyone took it yet, but we're going to talk more about the test. Overall, I'm pretty happy with the outcome, with the average outcome. Okay, today we're going to talk about single gene disorders. We talked about basic concepts like dominance, recessive alleles, compound heterozygotes. We talked about haplotypes, locus heterogeneity. So you see now these concepts, I understand that they're still vague because we didn't really talk about what, for example, a recessive genotype or dominant genotype means, how this is achieved on this molecular level. So this is what today's lecture, or two lectures actually, are about. I'm going to use some diseases, but I'm going to focus on one family. This is just to highlight the mechanism, genetic mechanisms involved that underlie these conditions. So you have a better understanding of a genetic disease in general. Because what we're going to talk about today doesn't apply only to those particular conditions. It applies to pretty much any genetic disease, including cancer and so on. So here's the definition. We're talking about genetic or molecular disease. So molecular disease is a broader term than genetic, because when we say genetic, we typically think about inherited condition, right? A molecular was introduced over 6 decades ago, and it refers to disorders in which the primary disease-causing event is an alteration, either inherited or acquired, affecting a gene, its structure, or its expression. So this is a broad definition, as you saw. It applies to across the board to many disorders, including cancer, autoimmune conditions, and so on. But first, you need a good understanding of the structure and function of a eukaryotic gene in general. And that's probably something that you saw before, if not here, elsewhere, but you had the basics of it in the biochem class, so it's kind of like a recap. And also talked about average human genes in the context of human genome. So you know that it's very important for a gene to be regulated in a very fine-tuned way, right? It's in concentration with other genes, and this is achieved through cis and trans regulatory factors. So when you look at this picture, it's convenient actually to divide this complex into two major components. Cis regulatory sequences belong to the DNA molecule on which the coding region is located on. So everything adjacent to the gene, like the core promoter, kata box, and so on, are sequences that are crucial for regulatory control of the coding region, of expression of this gene, right? So this is cis factors that, like I said, are basically sequence features are adjacent to coding region, but some of them actually can be quite far away from the gene that they regulate. That refers to enhancers and silencers. Those are sequences that respectively attract proteins called activators or repressors. So activators bind to enhancers. Once they do that, expression of this target gene is increased. Unlike repressors who bind to these silencer sequences and decrease expression of the gene. This is all quantitative, so it's hard to tell if this is like complete silencing. Sometimes it is complete silencing, but not always. Like it's really quantitative. It varies from gene to gene. Even for the same gene, it depends on the cellular stage, developmental stage, and so on. And of course, you see the major agglomerate here of proteins consists of RNA polymerase, but before RNA polymerase can bind to the gene, to the core promoter, there are other factors, other proteins that have to be in place, like this TAPA binding protein, all these basal transcription factors. So those are like really critical players in gene expression and gene regulation. So these BFEH are basal transcription factors, and this TATA box you probably heard about too. It binds to a very specific motif here, the sequence motif that is within the core promoter, and all these co-activators that are numbered mostly. They interact with TATA binding protein, with basal transcription factors, as well as activators and repressors, and all these composition, the repertoire of these proteins of the complex is quite dynamic, and that allows to regulate gene expression in a very fine-tuned way. So this is transcription that you should be aware of. So first, once transcription is activated, the primary transcript is produced. It first includes introns, then introns are spliced out. 5' cap is added, and these processes lead to a mature transcript of a mature messenger RNA, which then is translated into protein. All these steps, including splicing or even the transformations of the ends, like the formation of the 5' cap, are also regulated by other proteins. So all these proteins contribute to fine-tuning of gene expression. So that's the basics of eukaryotic gene structure and regulation, which are important for the next things that I have to say. So now let's talk about mutations and what effects they may have. So in general, we divide mutations into those that change proteins, they change the structure, leading to some abnormal protein, and those that preserve the structure, but they change the amount of the protein. So, which obviously refers to mutations that happen in the coding region, so those lead to changes in protein structure, and those that affect regulation or dosage. So those take place in encoding sequences, but non-coding regulatory sequences, like the promoter region. So let's talk about protein structure changing mutations first. And this arrow, its strength suggests that this is the major mechanism, and it is of course very important mechanism. But I wouldn't say that this is more common than structural. So this arrow is also very important, even though it looks thinner in this slide. But speaking of abnormal proteins, so now you may have various scenarios leading to it. The first one is when the structure is so changed that the protein is unstable. So it's like a dominant negative effect, right? It's, there's not much more to say about it. An example is hemoglobin in Hammersmith that I'll tell you more about later on. There are many mutations that lead to loss of protein function. So this is the majority of mutations in this under this class, which makes sense because if there is a sequence, functional sequence that is translated into protein, this protein is functional, it evolved over millions of years, it's obviously easier to break it than to improve it, right? So most of the mutations that will happen within the sequence will lead to some dysfunction, mostly loss of protein function. Sometimes the effect is opposite, which we call gain of function. That's relatively rare, but that happens. So for example, I'll give some examples later on, but basically the function is increased. So whatever the protein does, there's some extra activity associated with the mutation. And hemoglobin, ChemZ, or chondrodysplasia are examples of such diseases. Finally, there might be some completely novel function due to property due to the mutation. This is rare, but it also happens. And hemoglobin S, associated with sickle cell anemia, is a good example of it. These properties are not really beneficial, but they can be, even in the case of HBS, even though it is a disease. We're going to talk about it. There are some aspects of this disease or this mutation that can be beneficial, which is very interesting story. But there are more examples like that. Okay, so this is one side. This is the side related to abnormal protein structure. What about this protein structure normal? What does it mean? It means, again, that the structure is preserved, but the amount of the protein is either decreased or increased, or there might be some inappropriate expression, which means that the protein is expressed in the wrong time or place. So for decreased amount, there are some examples here listed. It's alpha thalassemia. Monosomies can be also treated as such. Tumor suppressor mutations. Trisomies, Charcot-Marie-Tooth disease type A, are examples of abnormal upregulation of a gene. So there's more product than expected leading to these conditions. And then inappropriate expression, again, that refers to time and place, refers to such conditions as hereditary persistence of FIDO, hemoglobin, and some oncogenes and so on. So I think I covered the slide. Any questions about it? So this is a very important slide because it basically summarizes the lectures for today. So now let's talk about recessive and dominant mutations. So how does it translate into molecular mechanisms? So let's assume that there is only one gene active, the other is mutated here, compared to this normal situation when there's 100% of normal protein amount and activity. So the mutation can be recessive if activity of the remaining allele is sufficient to support the function. Or if activity of the non-mutated allele is not sufficient to support the function, we call it haploinsufficiency or just dominant mutation. So haploinsufficient means that again, the remaining non-mutated copy is not enough to support normal function of the gene. That leads to dominance. So of course you can call recessive states haplosufficient, right? Because again, the remaining allele gives rise to sufficient product and function. So this is loss of function mutation. So the loss of function may have either dominant or recessive effect. So how is it possible? How can a mutation produce dominance? So here's an example, and this is another term for it is gain of function. So dominant mutation can be either loss of function or gain of function. So now I'm talking about gain of function, right? That refers to situations when there is this molecular biological process known as switchability. So the function of this protein is controlled by some ligand. So let's say that there's some ligand that is required to bind to the protein to activate it. Otherwise, this protein is not active. So this is why, so the ligand is on or off. That's why we're talking about like a switch mechanism here. When this mutation happens here, and in one of these alleles, let's assume that affinity to this ligand increases. So the conformation of the protein, the structure of protein changes so in such a way that interaction between the protein and its ligand is now easier. So that leads to some constitutive permanent binding. And some proteins will be on all the time. So the cell now has no means to deactivate, to suppress this protein, right? So this is the dominant mechanism here. And in this homozygous state, when both alleles are mutated, now all proteins will be constantly activated. And that explains why this kind of dosage effect that we see, that many genetic dominant disorders are lethal. because of this major dysfunction here. When it's heterozygous, there are some proteins, there are some normal proteins that compensate for this deleterious effect. But again, because there is no way to turn off kind of these proteins, that leads to a dominant effect. Does that make sense? What about dominant negative mutation? So this is a slide that shows you loss of function, which in this case is dominant. So you saw that there are some proteins that form like tetramers or octamers, like in the case of histones or hemoglobins that I'm going to talk about today. So there are like protein molecule partners that are required for the complex to function. So like all 4 units in this example are required for proper function. So what happens if one of these mutations is, one of these alleles is mutated? And this mutation here is shown as blue. So this is active tetramer, and when you now play with the combinations that are possible, you may have 16 possible combinations, right? And just a fraction of them is the right combination of all red units here. So those are the functional. Even if there is one blue unit there, one blue monomer, that leads to a dysfunctional product. Let's say that this just collapses, doesn't support the function that it's supposed to function. And of course, that applies to other numbers of the blue product, of the blue monomer. So when it's 2, there might be 3 of them or 4 of them. All these combinations are dysfunctional. So again, because only a small fraction of all these tetramers are functional, it's not enough to support the function, and that's why it's dominant negative mutation. And negative means that it's loss of function versus the previous slide that emphasized gain of function. So that leads me to hemoglobinopathies, which are the most common single gene diseases in humans. So that's one of the main reasons that I'm going to focus on them today. Between 5 and 7% of the world's population are carriers of genetic variants for clinically important disorders of hemoglobin. And these disorders are relatively well understood and may serve as a model for other genetic or molecular diseases, including cancer. So they belong to pretty much all the classes of genetic heterogeneity that we talked about. So we have some examples of allelic heterogeneity, which again is the occurrence of more than one allele at the locus, resulting in this condition. Examples are listed here as alpha thalassemias and beta thalassemias. Locus heterogeneity, this is association of more than one locus with the same clinical phenotype. And thalassemia is also an example. It can result from mutations in either the alpha globin or beta globin genes. And clinical phenotypic heterogeneity that we didn't talk about yet, but it's kind of self-explainable. It's the association of more than one phenotype with mutations at a single locus. So for example, sickle cell disease and beta thalassemias each result from distinct beta globin mutations. So like you see, these conditions are very diverse, and they encompass a variety of genetic mechanisms. So this is a hemoglobin molecule, which is, hemoglobin is the oxygen carrier in vertebrate red blood cells. Each hemoglobin consists of 4 subunits, 2 alpha globin chains, or 2 alpha-like globin chains, and 2 beta or beta-like globin chains. So this one is hemoglobin A, which has 2 alpha and 2 beta structures. Each subunit, each of those 4, is composed of polypeptide chain, which is a globin, and a prosthetic group called heme that contains iron molecules of iron pigment that combines with oxygen, producing this oxygen transporting capability of hemoglobin. So this structure is very well conserved through evolution, and this interaction between these monomers is known as tertiary structure, and like I said, this tertiary structure is critical for the function here. So any mutations that disrupt this structure will have some pathological effects. Of course, there are some sites that are more sensitive, that will have more deleterious effects, like the sites which are close to this prosthetic, this heme site will have more dramatic effects than some sites on the edges, just for comparison. So here's some of the genetics underlying globin genes, or part of it actually. But let's talk about alpha and beta genes that produce the globins that are then used to form hemoglobin. So the alpha-like genes are on chromosome 16, and beta-like genes are located on chromosome 11. In the upper panel that you see, the curved arrows refer to the switches in gene expression during development. So it's important to understand how this works. And the lower panel, it shows you development of erythropoiesis, which is the production of red blood cells in the human fetus and infant. So let's start with embryonic hemoglobin. So we have 3 types of them: HB Gower 1, HB Gower 2, and HB Portland. And you see that the expression first comes from the zeta gene. So zeta gene is in the alpha like complex on chromosome 16. So this is the order zeta, alpha 1, alpha 2, and alpha 1. And this order actually reflects pretty well timing of expression of these genes and production of the proteins respectively. So first, like Gower 2, Gower 1 and Portland are expressed from this zeta gene. And for For beta-like genes, we have the following numbers: is epsilon, gamma G, gamma A, delta, and beta. So all these genes belong to beta-like complex, but when a hemoglobin is formed, only one of them provides the product for the hemoglobin. And this is, I don't know if you can see it, but it's shown here. So for example, GOER1 for beta-like genes have epsilon, then GOER2 also has epsilon, and for power plant, it's one of the gamma genes. So embryonic erythropoiesis takes place mostly in yolk sac, and then liver after weeks like 12, liver also contributes and takes over the process. So this panel kind of matches this diagram. So think of this diagram as a map of the major sources of expression of each hemoglobin type. And then there is one fetal form of hemoglobin, which is known as fetal or HbF, hemoglobin F, or fetal hemoglobin. That basically remains there until the birth. And infants and adults have two types of hemoglobin, HbA2 and HbA. HbA2 tends to be expressed earlier, right after the birth, and it contains delta gene product as the beta-like globin. And this HbA is the predominant form of adult hemoglobin in the normal condition. Oh yes, there are tons of them, and we're going there. But this is the normal state, okay? Just to show you the globin genes and their expression patterns in time and place. So I didn't say that in adults, bone marrow is the main place for erythropoiesis, and this is where globins are produced. You have a question?
I have a question. So when you talk about, let's say, the hemoglobin F making alpha 2 and gamma 2, is this gamma coming from both G gamma and A gamma, or it could be like A
Yes,
gamma
from both of them. Although there might be some timing differences, so gamma G comes first, and then the position kind of reflects the timing of expression, but you can think of it as a mix of both.
Mix of both. Thank you.
You're welcome. So that's important. It's something that will be subject to tests, and it's important for understanding all these conditions that come with mutations that happen in these genes. So again, please pay attention to the curves. This is the alpha level when it's expressed, and what type of organs. Then gamma. Gamma is first predominant during gestation, and then it drops dramatically after birth, and beta slowly increases through development, and then it predominates in postnatal age. So those are the main things that you want to remember. So this is the beta globin locus. Sometimes we call it locus even though it contains multiple genes, so to be aware of it. But this is the complex of genes, and turns out that there is a very important regulatory region upstream of this region. It's 6 kilobases away actually from the cluster of genes, 6,000 bases upstream, known as LCR, or locus control region. And it's interesting because it may be deleted. So there's some instability in this region leading to a deletion, which then results in severe thalassemia. It's known as Hispanic thalassemia. It's even gamma delta beta thalassemia. So when this region is deleted, all these genes are silenced. Now back to the question about mutations. So you see that there are a variety of them. They may happen in different regions, in different genes, and it could be of various nature. So first of all, we may have structural variants like sickle cell anemia. So that kind of refers to the very first slide, one of the first slides that I showed you, with all these possible mechanisms of mutations with a major classification or distinction between those that preserve structure and those that do not. So this is about those that change the structure. So we call them structural variants. And they have some important clinical consequences, including hemolytic anemia, which occurs because the hemoglobin tetramer is not stable. It may lead to altered oxygen transport, which is due to increased or decreased oxygen affinity, or to the formation of methemoglobin, which is a form of globin not capable of reverse oxygenation, which means that it's in an oxygenated state all the time, and it doesn't release oxygen as it's supposed to. And then thalassemias that occur because they reduce the abundance of globin polypeptide. Most of these mutations impair the rate of transcription, or otherwise affect the level of encoded protein. And then we have, so this is about structures or structure change, and then we have thalassemia that result from decreased abundance of one or more of the globin chains. And finally, we have this HPFH, which is hereditary persistence of fetal hemoglobin. This is a group of clinically benign conditions that infer the prenatal switch from gamma globin to beta globin synthesis, which means that this gamma globin tends to show up even though beta globin normally is present in adults. Just like excess of these beta globins that are characteristic of fetal phases of development. Now, you should notice one of your questions should be, why is thalassemia in both places? So you can see that thalassemia is listed as structural variant, and then thalassemia is the main condition related to expression change when there is no structure change. So what does it mean? The answer is simple. Structural changes are not always independent of the expression level. So sometimes there might be a change in protein structure that leads to its stability and so on, which is translated into the altered abundance of the protein. So again, structural changes are not independent of expression changes or the expression consequences. And that's why thalassemias show up in both places here. Yes, but it just with a focus on clinical phenotypes, even though our true focus is mechanisms. But you need to be aware of the consequences. But then, of course, in the medical program, you will learn a lot about, obviously, about the clinical presentation of these conditions. So here you have major classes of hemoglobin structural variants. So you have variant class with their specific names, associated amino acid substitution. So by definition, these structural changes are due to mutations that change amino acid sequence. And then you have the clinical effects of mutations and inheritance pattern, where AR corresponds to autosomal recessive and AD is autosomal dominant. Maybe before I describe the table, let's take a 10-minute break.